Hello, welcome to Coursera, welcome to the California Institute of Technology, Caltech. My name is Henry Lester. I'll serve as the instructor for this course on drugs and the brain. We're going to start with two modules, each about 6 minutes long. These are introductions, and so there will be no assessments after these two introductory modules. I'll show you the kinds of images that we'll be using, the kinds of concepts we'll be using, and the kind of pace that we will be using. We'll be learning some pharmacology, of course, a bit of neuroscience, not so much as I would like to teach you, but there will be other courses on that. Some biochemistry, some biophysics, and of course, a little bit of disease orientation. So, let's get started with this first module, an overview of some basic science. First, we'll ask, what is a drug? And we'll give 4 examples. We'll give, as an example, nicotine, the addictive drug from tobacco. We'll be coming back to nicotine and the nicotine system fairly often in this course because it is the research of my laboratory. And I'm interested both in nicotine as an addictive drug and also in the fact that people who use tobacco for many years seem to have a lower, not a higher, but a lower incidence of Parkinson's disease. Then we'll talk about procaine during our first introduction to what's a drug, a local anesthetic, a synthetic one. We'll discuss morphine, a pain reliever, which is also addictive, during our first module on what's a drug. And during our module on what's a drug, we'll also talk about botulinum toxin. Botulinum toxin may be one of the first of the protein drugs that can be used on the nervous system. And we'll discuss its advantages and its many uses these days. Of course, drugs in the brain also requires us to talk about the brain. As we mentioned, I'll introduce just a bit of neuroscience. For instance, the relationship between the brain and the spinal cord in the central nervous system, some of the terms we use to discuss the brain, and some of the basic concepts. We'll talk about large-scale circuits of neurons, small-scale circuits of neurons, nerve cells, the contacts between individual neurons, the little chemical hop from a presynaptic neuron to a postsynaptic neuron. We call this little chemical hop the synapse, of course. I'll remind you that an adult brain contains about 10 to the 11th neurons, 100 billion neurons, and each of these might receive about 1,000 synapses apiece, for a total of about 10 to the 14th synapses. Nearly all of these form during the first 2 years of life, and so a fetus and an infant is very busy making about 1 million synapses per second. No wonder they get so tired. Then, of course, we will talk about drug receptors. I'll remind you that most drug receptors are membrane proteins. Here is our example, the nicotinic acetylcholine receptor. It has a region where drugs bind, a region that goes through a membrane, and a region that has that extends into the cytosol. Down here at the bottom of the slide is a link to a database at the NIH, the National Institutes of Health, which will give you a 3-dimensional structure that you can manipulate yourself to understand the basic parts of a protein and the basic structural themes. We'll be posting slides that have URLs on them on the course website so that you can follow these URLs yourself without any further work. In terms of drug receptors being membrane proteins, and again here the nicotine receptor, here is actually the best resolution we have of the binding, a nicotine molecule binding to its receptor. And then we'll go on to talk about the link between binding to a receptor and activating another part of the receptor. Here we will talk about activating a channel. Of course, we'll do this for several receptors in addition to this example, the nicotinic acetylcholine receptor. An important other aspect of drug action on ion channels is the fact that drugs block ion channels, in many cases literally like a plug in a drain. Here is a cartoon of a single channel recording. We'll mention single channel recordings briefly, and we'll discuss the fact that when the plug is in the ion channel, it doesn't conduct, but when the plug is out, the ion channel conducts as usual. We'll talk about how sometimes these plugs get stuck in ion channels, and that's the basis for anti-epileptic drugs and for anti-arrhythmic drugs in the heart. Having talked about ion channels and a little bit of electricity, we'll then go to an important part where we discuss drugs acting on G-protein pathways. The G-protein coupled receptor is a bit more complex as a pathway than ion channels. We have the receptor itself with 7 transmembrane helices. We have the G-protein. We have the effector for the G-protein. We'll spend some time discussing several aspects of this pathway because it has several different types of action in the brain. And then we'll talk about how to measure the intracellular effects of G-protein coupled receptors and of ion channels. We can measure some intracellular effects using advanced microscopy, live cell imaging, usually with fluorescence, and we can measure other effects using biochemistry. I'll stop here and let you absorb this material. I do want to remind you to see 2 items on the course website. The first is my sources of research funding. A professional would call this my disclaimers. And the second disclaimer is about medical advice. You should not take medical advice from me or infer that I'm giving you medical advice, and you should not use this course to give medical advice to any friend or family member. See you in a little while when we go to the second introductory module.